Hi, everyone. I would really like to thank the organizers for inviting me for this talk, and I'm really happy to have the opportunity to talk to you about my work. So I recently joined the Department of Computer Science at Virginia Tech as an assistant professor, and today I'm going to talk about my work on inferring cell lineage trees and differentiation maps from lineage tracing data. So I want to start this talk by talking about what I think is the most fascinating process in biology, which is embryogenesis. So this is a process which starts with just a single cell, which we call the zygote, and ends with a multicellular organism with multiple cells and multiple cell types. So there's really two processes that are going on here simultaneously. So the first is cell division that allows you to go from one cell to multiple cells. And then there's also cell differentiation that allows you to go from one cell type to multiple cell types. And the history of cell divisions is often represented by a cell lineage tree. So this is a rooted tree where the leaves are representing the cells that comprise the organism. And the root is representing the zygote and is showing you all the successive cell divisions that happen during development. And similarly, the cell differentiation can also be represented by a directed graph, which we call the cell differentiation map. And here the nodes are representing different cell types, and the edges are telling you what cell type transitions occurred during development. Now, these two processes are not just important for early development, these are actually happening in your body every single day. So an example of this is hematopoiesis. So in your bone marrow, billions of cells are being produced every single day. And here I'm showing you the differentiation map for human hematopoiesis. So you can see the hematopoietic stem cell at the root. The cell is dividing and differentiating, and you get this hierarchy of progenitor cell types. And as you go down this differentiation map, you'll see different cells that make up your blood. All right, so one of the central problems in developmental biology is what is the history of cell divisions and cell differentiation during development? So for an organism, what is the organism's cell lineage tree and what is its cell differentiation map? And some of you might be surprised to know that this problem is completely solved for some simple organisms. So one such organism is C. elegans, and the cell division history and differentiation of every cell of this organism has been mapped. And this was done using careful direct experimental observations in the 1980s. This is a very simple organism with less than 1,000 cells. And eventually this led to the 2002 Nobel Prize in Physiology or Medicine, because we learned a lot about gene regulation of organ development and programmed cell death. So imagine how much more we can learn if we could recover the history of cell division and differentiation for more complex organisms, perhaps for mammalian development. And now the direct experimental observations, the kind of approach that worked for this simple organism won't scale for large complex organisms with billions and trillions of cells. But now we are in the era of sequencing technologies. So I think this is a very exciting time to work in this area because of recent development of lineage tracing technologies. So in these technologies, you introduce artificial mutations in cells as they are dividing and differentiating using genome editing tools such as CRISPR-Cas9. And then you can use single cell assays to measure these mutations and the cell types at the leaves of the cell lineage tree. So with these lineage tracing technologies, you get two kinds of data. So the first is this kind of a matrix where each row is a cell and each column is telling you something about the artificial mutations, the presence and absence of these mutations as they were introduced. And you also get the information about the cell type of these cells. And it's important to note that you only measure this at the leaves of this cell lineage tree. And so now you need to use some computational method to recover the cell lineage tree from this data. So there are really two kinds of problems here. The first problem is, how do you go from this, what we call in phylogenetics, we call it a character matrix. Each column is called a character. So given this character matrix, how do you build the cell lineage tree? That is the tree that tells you the ancestral relationship between the cells. There's a one-to-one correspondence between the leaves of this tree and the rows of this input matrix. We call this the cell lineage tracing problem. Okay. And because you know the cell type of these leaves, this tree is leaf labeled with different cell types. The second problem is once you're given a leaf labeled cell lineage tree, how do you use that to build the cell differentiation map? So how do you tell what kind of cell type transitions occur during development? We call this the cell differentiation mapping problem. And these are the two problems that I'm going to talk about today. So for the cell lineage tracing problem, I'll talk about a method I developed during my postdoc called Startle. This was published in Cell Systems in 2023 and also accepted at Recom. And then in the second part of the talk, I'll talk about cell differentiation mapping. So this is towards the end of my postdoc. I worked on this. It's accepted in Nature Methods this year and was also at Recom this year. All right, so let's talk about cell lineage tracing. In this context, I'll be focusing on CRISPR-Cas9 based lineage tracing. So here, CRISPR-Cas9 is being used to introduce these artificial mutations. In certain targeted regions of the genome of these cells, CRISPR-Cas9 is used to introduce double-strand breaks, which when the cell tries to repair these damage to the DNA that has happened, it can sometimes introduce, it can make mistakes and introduce these insertions or deletions. And what we need to understand is that there are some unique characteristics of this process. The first characteristic is that these mutations are irreversible. So at a target location, once the CRISPR-Cas9 introduces an insertion or deletion, you cannot go back to the unedited state, okay? And the other important part is that these mutations are also non-modifiable. So what that means is once the indel insertion or deletion has been introduced, CRISPR-Cas9 cannot act on it further anymore. And so these are kind of stable that way and heritable. And lastly, we call it multi-state. So at each target location across cells, you can see different kind of mutational states. There are multiple states, okay? So for the unedited state, I'm going to call it state 0. Any of the edited states, I'll just number them 1, 2, 3, and so on. And let's see this process happening. So you always start from the no mutations. So all the target regions have state which you can measure, okay? So each column here is a target location. The entries are telling you the mutational state, and each row is a cell. And the measurements are only at the leaves of this tree, okay? So all of that was more schematic and cartoonish. This is real data. So here is what the character matrix looks like from, I pulled this data out of a 2022 paper on lineage tracing. So each column is a target site, and different colors here are telling you the different mutational states. And each row is a cell. So first thing you might is that there is a large number of states. So some of these columns have very many different colors. So there's a lot of states in certain target sites. It's not apparent from this plot maybe, but you can have hundreds to thousands of cells. So this is very high throughput data. And all the white regions here are basically missing entries. So there's a lot of missing information in this measurement. Up to 20% of entries can be missing. And because of these unique characteristics, a lot of specialized phylogenetic methods have been introduced to try to solve this problem. I won't go into details of those methods, but what we were interested in thinking about is what would be the most appropriate model for the evolution of these CRISPR-Cas9 induced mutations. So in the literature, we looked at some models which had some of the characteristics, but not all the characteristics of these of this process. So we developed a new model. The way we describe these models is using state transition diagrams. So these diagrams are basically directed graphs where nodes are telling you the different states and edges are telling you what kind of state transitions are you allowed to have in the process. So this is what we call a star homoplasy model because the state transition diagram is a star tree and it has all the right properties. So now that we have formally described this model, let's think about how we can use this model to build the cell lineage tree. So to do that, we use the what we call a maximum parsimony approach. So in this approach, you have certain weight associated with each state transition. It represents the probability of that mutation occurring. Okay. And in this framework, you want to, given the character matrix and given this weighted state transition diagram, you want to build a cell lineage tree that minimizes the total weight that is most parsimonious. And so we can pose this problem. And an important thing to know about this model is that the same transition can occur multiple times this is known as homoplasy. And homoplasy is what makes this problem hard. In fact, we showed that this problem is hard even if the number of times these mutations can occur is fixed, as long as that fixed number is greater than or equal to 4. All right, so this is a hard problem, and there are many standard methods in phylogenetics to solve these hard problems because honestly, most problems in phylogenetics are hard. And one such process is one such approach is using hill climbing. So you try to explore the space of trees, and you use certain moves to do this exploration. And one standard move that people use is nearest neighbor interchange. So we won't go into details of this, but to use this approach, you can think of a simpler problem. So given a tree, can you evaluate that tree? Given a cell lineage tree, what is its parsimony score given the weighted state transition diagram? And so this problem is much simpler. You can actually solve it in polynomial time. In interest of time, I won't go into details, but we actually found a faster way to do this. You can evaluate all the trees in the neighborhood of a given tree in time that is faster than if you would do it naively using standard approaches. All right, so we have this approach to do this tree search and try to build the cell lineage tree from the lineage tracing data. Let's look at what we can do on real data using this kind of approach. So here I'm going to talk about some data from a recent paper on mouse metastatic lung cancer. So here they had a mouse model where you can introduce this lineage tracing barcodes, and they were studying cancer in this mice. So one of the largest data sets they had from this paper was for a mouse where they extracted 21,000 cells from this mouse across 5 tumors in the same mouse. And these are the different tumors with different number of cells. And then the question is, what is the ancestral relationship between all these cells of all the tumors in this mouse? So the first thing we did is we built our phylogeny, compared it to the phylogeny that was published in the paper, and by design we are going to find a parsimonious phylogeny. So unsurprisingly, we actually do find a more parsimonious phylogeny than what was published. But then you can also do more with this. So here, each leaf here is labeled by the anatomical site from where the cell was extracted, from the tumor where the cell was extracted. And using this, you can try to understand what kind of migrations this cancer cells took during metastasis. And using our phylogeny, you're able to explain the data with fewer migrations than if you use the published phylogeny. So this gives some sort of evidence that maybe our phylogeny is closer to ground truth. All right, so now we have a way of building a cell lineage tree. Let's talk about the second kind of problem where, given a leaf-labeled cell lineage tree, how do you build the cell differentiation map? Now, this problem would have been very easy if we also knew the cell type of the ancestral cells. So why is that? Because let's say you do have a cell lineage tree where you know the cell type of every cell in this tree. You can build a cell differentiation map simply by observing the edges of the cell lineage tree. The source and the target node have different labels, and you can build a cell differentiation map that way. So that would be really easy, and I wouldn't be giving this talk right now. The problem is that we don't actually measure the cell type of these ancestral cells. All the measurements are only at the leaves. And that actually has 2 big challenges that we have to face. So the first challenge is that there might be some progenitor cell types here that are not observed at the leaves at all. So these are unobserved cell types. And the second thing is that some of the leaves might also be progenitor cell types themselves. So in this example, there are 2 progenitors that are not sampled, and 2 of the cell types that are observed are in fact progenitors themselves. So how do existing methods try to circumvent these challenges? So there are 2 large groups of methods for cell differentiation mapping in the literature. And one of them is, you can group them into, call them trajectory inference-based methods. So here you're using these cell type measurements or RNA-seq measurements of these cells. And you try to build curves or ridges in these gene expression space. But you're making certain assumptions here. And the big assumption you're making is that all the progenitors that are important for the cell differentiation process are all observed in your data. So that's a big assumption. On the other extreme, more recently, there's also methods that use distance-based heuristics to try to infer some sort of tree-structured cell differentiation map. And here you're making almost like completely opposite kind of assumption, where you're assuming that all the observed cell types are basically the terminal cell types, and none of the progenitor cell types are observed in your data. Okay. I would expect that, depending on the system you're looking at, reality might be somewhere in the middle, whereas the early progenitors are not observed, but the later progenitors are observed. Okay? And one important thing that we wanted to try to answer in this work was, how do you find the right number of unobserved progenitors, and how do you model these unobserved progenitors? So the key idea underpinning our approach is that we model these unobserved progenitors by their potency set. Okay, so a potency set is the set of cell types that a particular cell's descendants can attain. And this sort of formalizes how developmental biologists also describe progenitors. So, for example, the lymphoid progenitor is defined as the cell whose descendants can be different lymphoid cells, right? Okay, so now we have this representation of a cell differentiation map, where the nodes are either cell types themselves or sets of cell types, okay? And let's say that you do have a cell differentiation map, and you have a cell lineage tree where the leaves are labeled. How do you evaluate how well this tree fits with this cell differentiation map? So this evaluation can be done by trying to label the internal vertices of this tree using the potency sets that are given in your cell differentiation map. Okay, so this is a vertex labeling problem, and once you have such a labeling, you need some sort of objective function, some sort of way of evaluating how good that labeling is. So we came up with we came up with a metric called discrepancy. So this is we count the number of discrepancies in the observed potency, that is the cell types that are in the subtree, rooted at that cell, and the labeled potency. So this way, so here in this example, there's only one cell whose observed potency does not match the potency that is described here, and that's this cell, and the number of such discrepancies is 1. So discrepancy is 1, and the number of progenitor cell types in this cell differentiation map is 4. So what we observed is that if you only have one possible progenitor, it has to be this kind of cell differentiation map, and that will lead to a lot of discrepancies. Whereas on the other extreme, you can always minimize the discrepancy and make it 0, but then you will have a lot of different progenitors in your cell differentiation map, right? So there's sort of a trade-off, and this was the ground truth for this data set. So there's a trade-off between the number of progenitors and the discrepancy. So simply minimizing discrepancy is not going to work. So the way we pose this problem is, given an integer k, can you find a cell differentiation map with k progenitors that minimizes this discrepancy? So that's the problem that we pose. We study this problem in multiple different ways. We show that it's hard. We show that even counting the number of such cell differentiation maps is sharply hard. On the positive side, we also show that you can actually solve this. It's fixed parameter tractable on the number of cell types. And in typical data sets, the number of cell types is much, much smaller than the number of cells. So for a lot of real data sets, this is actually a good result. And so we developed this method. We call it CARTA. It allows you to systematically look at the trade-off between the number of cell types, the number of progenitor cell types in your cell differentiation map and the discrepancy or the fit of your map with the data. And let's see what we can do on real data with this. So I'm going to go back to the hematopoiesis example. So this is a data set from 2020 where they used lentiviral barcodes to introduce barcodes or mutations in these cells. So these are mouse hematopoietic progenitor cells. They introduce these barcodes at day 0, then they let the cells divide and differentiate, and they take samples at day 2, day 4, and day 6. And there are 5,600 different sort of barcodes that they were able to introduce. So you get a tree like this, 5,600 different trees that look something like this. Okay, total number of cells we are talking about here is 130,000 across 11 different cell types. And here I'm showing you, it's called a spring plot. It's similar to a U-map you saw in the previous talk. And these are the different terminal cell types, and the gray part over here are undifferentiated cells. So they do not belong to any of these terminal cell types. All right, so we used our approach to reveal this sort of trade-off between the number of progenitor cell types and the discrepancy of the discrepancy in the fit. And all other competing methods will always be, so we are getting what is known as the Pareto front. So all the methods will always be worse in either one of the two or even both the objectives here, which is the number of progenitors and the discrepancy. And there are methods that, so trajectory inference-based methods will just say that undifferentiated cells differentiate into differentiated cells. That's not really very useful for us right now. But in the published paper, they built a cell differentiation map that looks something like this. And this is, on the right-hand side here, is what we think of as the canonical differentiation map for mouse hematopoiesis. And on the right-hand side bottom is the differentiation map that our method inferred. And all the red dots over here are discrepancies between the inferred differentiation map and the canonical differentiation map. So as you can see, our method is able to more faithfully capture what we think should be happening in this data set. Not only that, you can also use this to try to predict the fate of day 0 or of day 2 cells in this data set. And so what we did is you can label these cells by what our method predicts will be the fate of these cells. And so that's what we are doing here. And there's a very good agreement between the gene expression of these undifferentiated cells and the fate that we predict these cells will have. So that also gives some sort of confidence that our method is perhaps doing the right thing here. All right, I tried to stick to the time limit. I'm 1 minute over, but yeah, those are the two methods I wanted to talk about. The QR codes for the paper and code are here. If you find any of this interesting, I'm happy to talk offline. And yeah, if you think these methods might be useful for your data, please reach out. Thank you.
Thank you for the amazing talk. I was wondering, in the second to last slide, I think you said a very good agreement for the day 0 undifferentiated cells. Mm-hmm. Do you have a fraction of cells that you have correctly predicted the performance for? So
we don't have the ground truth, way of telling which cell went where, because this RNA sequencing is a destructive process. So here, all we can really do is try to give some sort of argument based on the similarity in gene expression or some sort of marker gene expression. But so all the day 2 cells here are also leaves in our cell lineage tree. We don't know what would have happened if we had let them differentiate for a few more days and see what, where they would have differentiated.
In this data set, because they do some labeling,
yes,
you should be able to know the future fates for about 1,000 or so of the day 0 undifferentiated cells, because they have the same barcodes as day 4 and day 6. So you should be able to get, at least for those 1,000 cells, a rough.
So this is
ground truth.
Yes, I understand your question. So this is under the assumption that they only barcode cells at day 0. They only measure the cells at day 2, day 4, and day 6.
Right.
Okay. Now for the day 2 cells, maybe you can make an argument that day 2 cells, the gene expression has not changed as much yet, and so they can be a proxy for what the cells look like at day 0. And so we do that, and we see very good agreement. I don't remember the fraction off the top of my head, but we can talk offline about that.
Thank you.
Yeah. But we should be careful about this because there is variability. Even at day 2, the cells have changed a little bit. And so, yeah.
Maybe.
Thank you for the talk. Regarding the assumptions that these mutations are irreversible,
Yeah.
and they are constantly there, I was wondering what will happen if a cell experiences DNA damage, and the DNA damage repair machinery touches one of those sites that were edited by CRISPR-Cas9. The sequence, I imagine, could be removed by homologous recombination or non-homologous end joining.
Yes.
So are those scenarios considered in your framework, or the probability is so low that you don't care about those?
Yeah, so this is a very simple approximation where we don't care about those, yes, but there are more recent methods that are, and I'm working on extensions where I'm also thinking about those things as well, yes. Which, there's a pre-processing step where we try to avoid these kind of cases, and these are going to be a minority of cases, so most of the times things work out pretty well with these technologies.